In 1859, Charles Darwin described to an astonished world precisely how it worked. Species, he said, came not from God, but from each other in the process known as evolution. It was not a popular idea.
Is it credible that all favorable varieties of turnips are tending to become men?
This is the law of higgledy-piggledy.
We cannot believe that God wrote on the rock one enormous and superfluous lie.
In 1973, Stanley Cohen and his colleague Herbert Boyer showed how evolution could be engineered. They got the same reception. After splitting the atom and exploring space, gene splicing is the third and most controversial frontier of modern science. Certainly it is the least understood. It promises to transform our health, the food we eat, the world we live in, even ourselves, by cutting up and redesigning the very stuff of life. Its opponents fear we may rue the day we heard of it.
Genetic engineering raises the most fundamental issue ever to face the human family. Do we engineer the genetic code of microbes, plants, animals, and humans to the short-term commercial needs of the marketplace? Do we become the architects of the blueprint of life over the coming decades and century? The resolution of this issue will go a long way to determine the kind of future that we will provide for our children's generation.
The variety and abundance of life in the Galapagos were the inspiration for Darwin's epic-making insights on evolution. But his theory of evolution was just that. Of its actual mechanism, Darwin knew nothing. Over the next 100 years, that will be charted too, setting the stage for the life revolution that genetic engineering would bring about. But the voyage of discovery was to run a crooked course. Cells had been known about for hundreds of years. But just as Darwin sailed in 1831, a smaller unit within the cell was noted. Called the nucleus, it fast became the focus of scientists' attention. In the 1860s, Friedrich Miescher isolated the material from the pus on bandages. A nucleus was made partly from complex proteins and partly from a simple chemical compound, which he called nuclein. Deceived by its simplicity, Miescher studied it for 30 years, never believing it held the secret of life. We call it DNA. Fashionable folk of the 1880s decked themselves out in brighter colors, made possible by the new chemical dyes. Biologists like Walter Fleming found other uses for them. Some parts of a nucleus soaked up the dye more than the rest. These chromosomes, from the Greek word for color, were seen to duplicate themselves as cells divided. It was a microscopic barn dance of startling significance. Rapidly, each animal species was found to have a characteristic number of chromosomes. All of its cells contain that number, except sperm and eggs, which carry half the number each. Proof enough that chromosomes were indeed the mechanism of heredity. As the Victorian age was laid to rest, so also were many old ideas about the way life worked. Ironically, though, the first great influence on the genetics of the new century was an Augustinian monk whose discoveries had gathered dust for years. Gregor Mendel was the father of modern genetics. In 1865, he showed precisely how traits are passed down through families. Mendel's laboratory was a monastery garden, and peas the subject of his experiments. His discoveries were seized upon in a new century which knew the extraordinary differences between individuals could not be accounted for by just 46 chromosomes. Some smaller unit of inheritance must exist, a unit that came to be called a gene. Many of the groundbreaking discoveries of the next decades were made by Thomas Hunt Morgan. Studying the fruit fly, he proved that chromosomes were indeed subdivided. By the 1920s, the large team gathered around him showed that the tiny fly must have at least 2,000 genes. At Columbia University, and with summer school students at Woods Hole, Morgan demonstrated how it was that certain traits are linked, like blonde hair and blue eyes, why some traits are linked to sex, and why roughly equal numbers of males and females are born. Why not breed better people? In the teens and 20s, eugenics became the new craze. At special exhibitions, there were trophies to be won. Best couple, best small family, best average family, best large family, even best girls club. But there was a serious side to all of this. Rules learned from peas were applied to people. Why take such care to breed livestock while leaving our own pedigree to chance? Mental patients would be sterilized in 27 states because, while all were equal in the land of the free, some cost more to maintain. Liberty continued to welcome the tired, the poor, and the huddled masses, but selectively from 1924. Vigilant eugenicists saw connections between race and intelligence. At a time when statistics show the average American had a mental age of 13, applicants from Southern and Eastern Europe were not considered bright enough to breathe free. People rallied to eugenics in Germany. The peasant who prevents weeds enables the valuable crops to grow, said the propaganda. Only the very fittest families were welcome in the 1,000-year Reich. And a society which valued uniformity above all else quickly weeded out the eccentric, the mentally ill, inherited disease, criminals, and, as an afterthought, Jews. An officer of the American Eugenics Society said, Hitler's sterilization policy showed great courage and statesmanship. he was not to know how it would end. The game had changed. This time, there were no trophies. From that time on, the word eugenics hid in shame. The specter of what happened still haunts genetics. In 1944, Oswald Avery, a quiet Canadian, finally proved that the hereditary material was not protein, but DNA, although his achievement was rapidly overshadowed. This is how the new atomic physics announced its arrival. But biology was catching up fast. It took 80 years to find out what the molecule of life was made of. To discover what it looked like would take just 8 more. The calm surface of Cambridge was ruffled in the spring of 1953 when a great scientific race was won by 2 of its junior scientists. James Watson, a 24-year-old American, together with an English refugee from physics, Francis Crick, announced that they had unraveled the secret of DNA's structure. A modest article in Nature magazine included the first drawing of the now famous double helix molecule. They had painstakingly constructed a wire and cardboard model, ignoring instructions not to waste time on such research. 1 Nobel Prize and a distinguished career later, James Watson still recalls those days with relish.
It was that period after the war when the English hadn't given up. That is, they wanted to think they were still equal to the Americans, at least in the sort of problems they might choose to work on. Francis is very sensible. He used to think I was silly. I wasn't, but he would talk so much sense that we would get overwhelmed by it, and so we would talk nonsense back to him, and he thought we really believed it. But it was just to, if you're overwhelmed by intellect, sometimes you just have to pull his leg, and sometimes he didn't realize that we knew we were just being silly.
And this is what came of all that inspired silliness. Imagine you could slide down DNA from one cell in your body. Like this, it would take 30 years. But for all its great length, it is simple. 2 serpentine backbones linked together by just 4 other chemicals called bases. But we will see, and this is the heart of the matter, each base fits with only one other. For now, think of those chemicals as letters. A group of them makes up a gene. So your DNA is a message, and what it describes so simply is you.
Now, looking back at it, I think the only answer we could have found was simple, because you had to have a way when life started for the genetic material to be copied. And if that had been too complicated, it would never got started. So if life arose spontaneously 3 or 4 billion years ago, you might have guessed, well, there was some simple chemical trick which allowed for genetic information to be replicated, and that's what we saw.
The trick works like this. We saw how each of the 4 chemical bases in DNA, called A, C, G, and T, will join with one, and only one, of the other three. When cells divide, the links between the bases are broken, making two single strands. Molecules of the four base chemicals at large in the nucleus are attracted. Because A only fits with T and C with G, each single strand must end up an exact replica of the original DNA. At last there was an explanation of how cells were copied and how genes and the traits they carried were passed on faithfully from generation to generation. But learning the language of DNA also shed light on evolution, because ironically, that language was sometimes imprecise.
We knew that they couldn't be 100% accuracy. In fact, initially we worried of how the replication scheme could be quite accurate enough. And now there are phenomena called DNA repair, things to make the replication more accurate than it must have been in the beginning. But sort of mutation was an inherent element in it. The replication scheme would never be perfect, and when it wasn't perfect, you've got a new form of genetic material, mutation.
Evolution does not proceed in straight lines, but by a series of mistakes. Some mutations give certain individuals advantages and become part of their species makeup. All tortoises can feed at ground level, but arching shells help in reaching food at greater heights. Land iguanas have round tails and a pointed snout. Flat tails propel their seafaring cousins through the water, and their snub noses are ideal for grazing the rocks. These were some of Darwin's conclusions, and a century of discovery proved him right.
We really now know what evolution is based on, changes in DNA molecules. So the real uniqueness of man is DNA in some senses. It gives us our extraordinary brains and has let us develop the languages and develop these cultures that we so admire. It's our DNA. So what we are, it comes, not from the heavens, but from the evolution of DNA.
The DNA held together by...
DNA is also important because it is the blueprint from which all proteins are built.
Now what happens when we have protein synthesis?
Most of our body is protein. Muscles, brain cells, hair, and hormones. What would once have won you a Nobel Prize is taught in school today.
Single strand, then is copied into what we
How
call
each
the
section of DNA, what we call a gene, contains the building instructions for just one protein. How the protein is made, and how DNA exercises control over life. So biology swung into the 60s, sure of the role DNA played in evolution, inheritance, and in building and maintaining living things. But in the age of Aquarius, another kind of science stole its thunder once again.
We choose to go to the moon in this decade and do the other things, not because they are easy, but because they are hard.
Biology had not stood still, but the big breakthrough came almost by chance. Antibiotics, the wonder drugs of the post-war years, had become less useful as bacteria grew resistant to them. It was first thought this happened by a gradual process of mutation. But then it was discovered that resistant bacteria could transfer this ability to others overnight. It was the role genes must play in this that interested Stanley Cohen.
It had been known for some time that many bacterial cells have, in addition to their chromosome, which has the bulk of genetic information of the cell, little pieces of extra DNA called plasmids. And certain of these plasmids carry genes for antibiotic resistance. We were studying how these genes made cells resistant, and in order to carry out some of our work, we wished to be able to take plasmids out of cells and introduce them into cells that lacked the plasmid.
By 1972, Cohen's team could remove from bacteria the ring of DNA, the plasmid. And they found a particular salt solution that softened the outer wall of other bacteria and let the plasmid get inside. At the same time, other investigators were becoming interested in chemicals called restriction enzymes, secreted by bacteria to destroy their own parasites. Restriction enzymes had the ability to cut up DNA. One of the experts in this field was Herbert Boyer. It was in Hawaii that the historic meeting of the two took place. One afternoon at a conference, Cohen heard Boyer describe a restriction enzyme called EcoR1.
That evening, after the session, Herb and I went for a long walk. We ended up at a delicatessen across from Waikiki Beach, and in the midst of eating a corned beef sandwich, I proposed a collaboration with Herb.
Cohen's insight was this. Taking one of his plasmids, why not use Boyer's enzyme to cut it? Restriction enzymes always slice DNA at a particular place, but Cohen saw EcoR1's advantage. It left overlapping ends like a joint.
Since all of the overlapping DNA ends produced by this enzyme were identical, it occurred to me that it might be possible to take plasmids and cut them within this enzyme and then link a segment of DNA with these same overlapping ends to the originally cut piece.
And that is how gene splicing was born. Using enzymes as scalpels, genes from one strand of DNA could be inserted in others. More enzymes were used as stitches, and the new gene worked perfectly in the new bacterial home. In fact, only 1 in a million bacteria took up the extra gene. But how do you find them? The original problem provided a solution. The genes Cohen spliced made bacteria resistant to antibiotics. The bacteria were placed on an antibiotic gel. If any survivors were found, the experiment was a success.
We were able to find such cells on our plates, and they could produce colonies, and in this way we were able to clone reproductions of the original composite DNA molecule that was made using this procedure.
The experiment did not stop at engineering bacteria that nature never created. Within a year, Cohen and Boyer would show that gene splicing worked its awesome power in much more complex animals.
Very shortly after that, we took the DNA from the frog, the South African frog Xenopus levis, and found that we were able to propagate animal cell DNA in bacteria. It was a very exciting time in the lives of all of us, and we were delighted, of course, with the possibilities that opened up.
But those possibilities did not delight everyone. Many were quick to perceive the revolutionary impact that gene splicing could have on life.
Evolution proceeds by mutations and within a species in sexual recombinations and so on within that species. And the genes certainly only very rarely, if ever, are transferred from one species to another. That's not been so that's why evolution proceeds, as we know, in a sort of a branching pattern. And what we were about now was to start moving genes from any species to any other species. In other words, to convert what has been a branching pattern into more of a network. And that evolution from now on would be different from all that has taken place in the past.
Others feared that dangerous new organisms would escape from the lab, an uncontainable new form of pollution.
An oil spill may be nasty, but sooner or later the thing decays away or you can wipe it up. I mean, it doesn't just continue to, to grow. But organisms, that's very different, right? You can have a very small release into the ecosystem, and if they find a little niche, they can establish themselves, and you can't pull them back.
As the debate heated up, and it seemed possible that governments might ban gene splicing altogether, scientists decided to regulate themselves. In 1974, an open letter called for caution until the dangers could be assessed. Cohen, Boyer, and James Watson all signed. Rules controlling laboratory procedures were established, but not everyone was reassured. On the very day the rules were published, a council meeting in Cambridge, Massachusetts, erupted in anger over a plan by Harvard to build a gene splicing lab in the city. In the end, the lab was built, but many sympathized with Mayor Vellucci's fears that sightings of orange-eyed beings and hairy 9-foot creatures may be linked to genetic experiments. The truth was less fanciful and more marvelous. Using bacteria to make millions of identical copies, or clones, of a gene, molecular biologists set about unlocking the secrets of life, and mutants are playing a part. This is a normal fruit fly, its 2 wings folded. This mutant has an extra pair, but it can't fly.